I'm Zev Wisotsky. I'm a product marketing manager at Revvity Signals, and I'm excited to share my presentation with you all. Before I begin, though, I just wanted to call out two assumptions that we are making with this presentation. So one, that we all have a basic understanding of antibody-drug discovery, and the need for digital tools to support this process is very important. I would almost say paramount to the drug discovery success. So just a quick background, at Revvity Signals, our mission is innovating for a healthier world. We're really working to fulfill that mission by providing science-based software solutions that leverage big tech innovations to help improve lives everywhere. So really what we say to people, in other words, is we make scientific software that help our customers to make drugs that save lives. Okay. So here is a slide of what we'll learn today. I won't read this from the slide, but I'm excited to introduce you to Revvity Signals. Our organization is focused on delivering drug discovery and development software to help accelerate your science. Before we get to the main part of the presentation, I just wanted to introduce our Signals Research Suite. We use a make, test, decide paradigm, and you'll see that in our presentation today, to support our researchers. So using Signals Notebook, the premier cloud-based electronic lab notebook that facilitates the creation and communication of molecules of interest via hierarchical editing language for macromolecules. You'll see that, Helm, and powered ChemDraw as well. Test, we'll use Signals Research Suite data processing for data analysis and visualization. Curve fitting, bioassays, and standardizing your results for consistent results, powered by Spotfire. And then finally, the Signals Research Suite data-driven analytics, which enabling multi-assay, multi-study comparison powered by Spotfire, and the ability to find the best candidate in whichever space you're in to move toward accelerating this decision-making. So specifically today, researchers will gain a better visibility into antibody-drug conjugates or just antibody therapeutics, candidate profiling, and can quickly determine, for example, structure, activity, or structure property relationships across modalities to select the optimal, in this case, conjugation sites and parents. Okay, so today's, just to put into context, today's drug hunters working in drug discovery and development have a tough job. They're looking for cures within a space that's getting increasingly more complex, running the gamut of traditional small molecules, which you can see on the left here, all the way to larger drug modalities up to cells, and we'll talk about antibodies today. Just to look at the market conditions, sales in the top selling drugs for at least 2022 was around a 40-60 split between small molecules and larger, more complicated biologics. And while the potential for patient health is quite vast, the company facing drug hunters is increasingly over the spectrum for small to larger, so more complicated biologics. In other words, new drug modalities are harder to represent, they're harder to make, they're harder to deliver to biopharmaceutical research organizations. And then adding these modalities together, I use the term like Franken molecules. So for example, an oligonucleotide conjugated to a peptide, or in our case today, we'll talk about antibody-drug conjugates. It increases the need for these tools to be more sophisticated, enough to support effective communication and increasing the speed and accuracy of developing these candidate drugs. So meeting the challenge of greater complexity means the next generation of informatics platforms has to cover this full spectrum. And at Revvity Signals, we're keenly aware of this complexity for new drug modality research, and have developed what we believe is the next generation discovery informatics platform to address these growing challenges of new drug modality research. Okay, so again, to make software powerful, it needs to communicate and represent science with the end goals in mind. For example, if you're working on small molecules, as a chemist, you'd require chemical structures and other tools to showcase how structures are made. As you get larger, working with a scientist potentially on oligonucleotides, you need consistent and easy ways to represent or build these, such as Helm or other sequence-focused tools. And as you get to the protein level, a scientist could need things like AlphaFold or other 3D structural tools. Although today we're focusing on antibody-drug conjugates, it's challenging to find software tools to connect and support the needs across these modalities. So for those who are unfamiliar, antibody-drug conjugates are generally made up of 3 different components: antibodies, which must be generated and bind to targets of interest, linkers, which can be chemical or peptide-based, and the small molecule warhead or the drug acting on the target. And as I mentioned, this really to note that we're already starting with potentially 3 or 3+ ways of representing individuals' work within many softwares, and this puts communication and information as a key requirement, which can be challenging to accomplish. So furthermore, I'll introduce you to our cast of characters today. By the way, everything you're seeing here is a faux biopharma, so this doesn't exist, but it's helpful to walk us through, and we work with many companies in this particular situation. So Hopper Biopharma is our company today, and working on antibody-drug conjugates also introduces scientific specialties in which potentially 3 or more separate subject matter expert groups will need to work together, making these final candidates for testing. So here's our team. So we have Alice in the top left, who's working on antibody-drug discovery team. Alex and Stacy's team will be on the assays. So one will be doing like primary and secondary screening. And then there are decisions that are made to either advance or re-immunize. We'll see in a second. Paul is in the protein engineering team, and Charles is from the small molecule team. So again, I'm saying this a couple times because I think it is easy to say software will solve everything, but we have to really think about, the people involved and who are doing this. It's a challenge to work seamlessly together. We're using various different tools, potentially disparate, not connected, not easily searchable. We have 3+ data work streams and data sets being produced, in some cases simultaneously. Some are highly dependent with no and go no decisions for gating various different aspects of the organization. And the ability for these groups, for yourselves, for people who are studying and working in this space, to be efficient, to fail quickly or to succeed quickly with candidates is a key metric for drug discovery. So let's review the ADC research flow, and then I'll show you how this looks and it is accomplished today in the Signals research suite. Okay, so we will first start with the immunization, harvesting of cells. We will then generate the hybridoma cell lines for testing in primary high throughput screens. In this example, maybe like an ELISA or others. Excuse me. Then we'll send for a secondary screening. We'll be using surface plasmon resonance assays, but there are other binding assays that one can do. And then from here, we'll go to our first go or no-go decision, where we'll make a decision on whether to start another immunization or press on. Assuming that these candidates meet the criteria that we're looking for, candidate antibodies will be selected for further optimization and expression, binding, humanization for antibody-drug conjugation. And then these antibodies are purified, and we'll need to combine the payload and the linker to generate the ADC, which will then be tested extensively, and finally to the ADC candidate nomination. So typically, this is much more complex, as you can kind of see here. So we've worked with companies, and this is an example. This process can take months, up to years, and it's more complicated than the previous slide that I represented. But to make I'll just mention that we work with many companies in this space. So to make our presentation a little bit more interactive, we will now post our first of 3 poll questions. I'll give the next 30 to 45 seconds to participate. So I think everybody should be hopefully seeing a poll. And then Janice, I'm not sure if you're on the line still to give us a time check.
Yeah, can you hear me?
I can, yes.
Okay, excellent. It still says mute on my end for some reason, but I think
we hear you great.
We're getting there. We may have all the answers in. Yes. Oop, nope, one more coming in. So we have yes coming in at 38%, no, at 31% and not sure at 31%. So basically a fairly even split here.
Yeah. Yeah. So the question was, how do you feel your current software supports antibody drug discovery today? And this is quite typical. We see kind of between a 25% to 30, as you saw here, or 32% of customers saying, yeah, we feel like it fits what we need. And then about, 2/3 to more, sometimes 75% in the I don't know, or for sure, no, it does not support. And that's definitely, so we appreciate your participation. And, one of our goals is to really support that and hear that voice and try to continue to make our softwares better. So appreciate that. We'll move on. Okay, so here's our outline. We already talked about the background. We'll first, discuss the discovery of antibodies of interest, the engineering team creating linkers and payloads of interest, conjugating and tracking binding and linker development via Helm. We'll integrate, and you'll see combining the 3 major components for testing for cell killing assays and other assays. And then finally ending with the full team to decide which antibodies are we going to promote. But instead of kind of starting from the beginning, I like to actually start from the end first to show you where we're going, so you guys are, very well aware of our end goal. So what we are looking at here is a part of our product. This is an example of a Signals data dashboard powered by Spotfire. And each of these columns is various data that we want to compare over many different candidates that are tested. So for example, we have small molecule payloads. You can see on the left, we have linkers or antibody binding sites and assay data all associated. But what we really want to look at is a dashboard that looks like this. Again, this is another dashboard powered by our data analytics. And this dashboard is all of the data that was assembled over various months for each constituent of an antibody drug conjugate, as well as the antibody drug conjugate data. And overall, if we focus on the green sort of bar or bounds, this represents the range in which we can compare candidates over many assays, many parameters, to be deemed effective or safe or good, or that fall within sort of what we care about as far as our antibodies that we're generating. So this allows us to compare various metrics and assay data, which candidates to move forward and why. And this is the end goal, right? This is what we want. So it's for our drug pipeline. So I can show you an easy way to compare this, whether you're a scientist or a VP of research or director or whatnot. I show you this because now I'll share the destination. I share the destination where our presentation will take us today. Okay. All right. So we're now in product. It first begins with Alice. She's the antibody scientist who is going to go to the vivarium. She'll stimulate immunogenicity in the animals. And Alice uses Signals notebook, right? That's where we're at, to prepare the animal experiments, recording protocols, including all of the objectives and the steps that go into, this. One of the great strengths, of course, is that it provides you an environment in which all of the inputs like media, animals, proteins can all be tracked and identified and linked together in a single experimental record. And it also provides a place where you can begin recording observations. So in this particular case, we want to look at titer of the antibodies. And this is integrated with Spotfire directly. So those observations can be directly linked and loaded, which we'll see in a second, into Spotfire and visualized, as well as example plate layouts and samples that are being used. So this is all now part of a single experiment that is supporting all of the workflows that, for Alice, that she would need to generate this for the animals. Okay, so the benefit or value for Alice is because Alice is responsible for multiple experiments, potentially in parallel. She has she needs a single environment in which that she can house all of those, each of those studies, and can more easily be tracked. All of the data for them, and it's all organized. Additionally, communication and the ability to share this data and materials is also very paramount, as her hybridoma cell lines will be needed for testing in the next stages. Okay. So moving on, next, Alex, the assay scientist, will take these clones that have been selected and run them through a battery of assays. Again, Signals notebook will assemble the media, materials, equipment, and protocols that will be used for his high throughput plate screen for primer activity. And so we will bring all of these results into Signals Notebook. You saw that in drag and drop, but this could also be done programmatically. And then Alex will use Signals Research Suite data processing experiment, again, this integrated version of Spotfire to support the high end-to-end process. And because all of the data is integrated for Alex, he can track and centralize the data processing. So he's doing a high throughput ELISA. The raw data comes in from the Signals Notebook. All the experimental properties, plate maps you can see here, and experimental details are being brought in. And then he will go to the high throughput sequence QA/QC app, which we'll see very shortly, and facilitate the normalized view of all of the controls, the plate layouts, and the results will be normalized. So that's what we're looking at here. And finally, this will be put into a hit selection, a little bit more. Still normalization results. So next we'll go to the hit selection, where put a little more simply, is where 4 colonies will appear for the hit criteria of interest and can be directly selected and sent back to the experiment or shared with the team for next steps of processing, queuing, and any updating of tasks, which is what we're seeing here. So we can now do that, the downstream work. And here's the cloning task. Okay. So the value for Alex is the ability to capture mass amounts of data seamlessly, as he has to screen thousands of these colonies, and having to process whereby he can drag and drop results in, although I mentioned the majority of that might be pushed in via APIs, have the experimental record, keep track of everything, and has automatic workflow that guides him through a step-by-step process and task requesting for secondary screening. Okay. So in our next scene, the team will run a binding affinity assay, such as the SPR experiment. So Stacy is an assay scientist who will take these antibodies and do SPR assays. We have the capabilities within our platform to run SPR processing from raw results to final data sets across different instruments. And in this example, she's bringing in the raw data from a BioCore instrument and then walking through the other Signals Research Suite data processing workflow. So the raw data is viewed here, and it goes into the normal steps performed in SPR, including zeroing, where we can select the part of SPR curve and everything will be baselined against. We'll be doing, I think, cropping, which is showing next, where we can take the full SPR flow cell and focus on the subsets of space that actually have the data of interest. Then we'll be doing referencing so that each of the flow cells can be correctly referenced to its reference flow cell for intra flow cell data visualization, and then normalized or blank subtracted to normalize correctly on an empty flow cell data to get the final results, which is, I think, coming here, blank subtraction, yeah. So finally, the data is put into a kinetic modeling, one-to-one mass transfer calculation, and we'll be doing a curve fit to be able to model the data and extract the right parameters. So modifying, the way the curve interpretation is being done, and finally getting the kinetic curve here, we're able to look at the Ka and Kds, which each of these values or results is useful for the team. And at this point, we can do a QA/QC for knocking out various different points of that are outside the bounds of interest. And then, of course, she can publish this data. That's what we're doing here via the publishing app, which directly pushes this into the data repository layer, to be integrated with other results, and then assess for which of these antibodies will be chosen to move forward, which is what we're doing here. So I know that's a lot, but really, for Stacy, the benefit is that this is an integrated platform and it allows for standardization and normalization of her data to be managed. As some of you know who work in this space, there are various different SPR instruments, right? Each one has its own different user interface and different user experience. But when you use Signals, she and her team have a common experience platform to support that secondary analysis, supporting data normalization, as I mentioned, workload, et cetera. So regardless of where the data is coming from, once that protocol or workflow is generated, her team only has to learn really a single interface for that secondary analysis, and she has the confidence that it's being processed the same way regardless of which user this is. Moving on. So having generated the hybridomas and generating antibodies, the team is now ready to decide which of these antibodies are most efficacious given the requirements. So we're looking at an antibody dashboard here to do so. And here you can access the global search where antibodies can be selected. Here we can see the individual's results for CDR regions in each antibody in just a moment. So that ends, so instead of like a SAR table for small molecules, we're using a sequence, SAR analysis. A lead antibody can be pinned and compared to the remaining antibodies to look for patterns of mutational differences between lead, ABs, and other antibodies that are being addressed. And at this particular example, we're looking at hex cell titer to select the lead antibody of interest. It's easy to compare for the sequence differences here. But easier would be to use the multiple sequence alignment. So moving to the multiple sequence alignment app, the scientists can pin the reference antibodies to compare, driving at the comparison where you can see the distribution of results. And again, you pin a single reference and you can look at the various CDRs that are in red and compare which mutations from lead antibodies, looking again at hex titer and other assay results. So you can see that being moved here. But even better would be to have a system return you an unbiased fashion where the best antibody by CDR region and within the antibody component analysis, the user can really select those shown and for improvement and of assay of interest, in this case binding or hex cell titer, and select the reference to see the sequence patterns that are really driving these results of interest. So the best antibodies are then selected at this point. They're sent back to the Signals environment for nomination moving forward with the ADC assembly. And this group list is available from here inside Signals analysis object, where antibody results can be reviewed with the team and how they decide to progress with the next steps of the overall project, which you're seeing here. Okay, so really, again, what is the value here? Identifying the antibody substitutions, correlating these to binding and titer results, and picking the best antibody efficiencies to move forward. We can identify which antibodies to advance. This and Signals Research Suite helps surface these results by integrating over various data sets, providing antibody relevant tools to make the process easy and as transparent as possible. All right. So we're going to take a quick break here. Here is our second of the 3 poll questions. Let's give another 30 to 45 seconds to participate from our audience. And maybe we can read the question as well.
Yep. So the question here is, you are generating scientific findings, and we would like to know how many teams receive your scientific findings. Is it 1 team that you communicate with? 3, 2, 4, more than 4? And answers coming in are favoring 2 at 53%.
Yep.
Smaller numbers are for 1 and 4 plus at 13%, about 20% at 3.
Yeah, perfect. I'll comment on that. I mean, I think that's definitely in line with what we see as well. Generally, you're working with more than 1 team, we'll say. And I think the goal with this is really to identify with the challenge of communication as you increase the number of teams that you have to interact with, and multiply that by the number of softwares you all use, email, et cetera, and meetings. It's a huge challenge. And so we really want to think about that as we generate software to make it more useful and what I'll call a joyful experience, which is hard to say with software. So appreciate that participation again. All right, we'll move on to a couple more scenes, and then our final poll question, and then open it up for questions. So let's we'll move on here. Okay, so having identified the leads, next we want to optimize expression or maybe humanization. So Paul is the protein engineer who will take the antibody sequence and put them into expression vectors, which we have capabilities within the Signals notebooks you can see here. Very importantly, Paul is able to do his cloning directly here. We have integrated tools like SnapGene But so this is very easy for molecular biologists like Paul. But in this particular example, we're looking at the molecular biology toolkit within Signals capabilities. So he can plan primers, he can develop a whole cloning experiment. So this is just, again, more sequencing here. And here we can plan primers, develop a whole cloning experiment, and allow Paul to easily create expression vectors for production. So within the Signals notebook page, you'll have the ability to track all of the constituents, like the original plasmid, primers, insert sequence, and so forth, and all the way to the PCR gel to verify that it's correct within the notebook to show the correct plasmid has been created. And then he can use now the worksheets within Signals notebook to track the production experiment, including steps that have been completed, timestamp when they've been completed, which really helps and guides Paul through the process of cloning, through expression, through purification. So for Paul, the benefit that Signals provides is really to make it easy for the molecular biologist to sit down, plan out the experiment, and the capabilities like worksheets to track progress, driving experiments for consistent, reproducible, and in a reliable fashion. We don't have time today, unfortunately, to go into the small molecule development, although I think this group may be the wrong group for that anyway. But we do have another presentation. I just wanted to mention that this is showcased. So if you're interested, feel free to reach out to me and I can share that with you. Okay, so moving on to Charles. He's responsible for the linker. The linker for this particular organization is their special sauce, and what differentiates them from other antibody drug conjugate companies. So Charles needs to have an easy way and a consistent way to draw these polypeptides or small molecules or intermediate sized molecules. And to do that, he'll need to create new synthesis experiments, which you're seeing here. And as I mentioned, this is a huge differentiator for them. So integrated into Signals is the Helm monomer editor, which you can see here. And it allows to support natural and customer curated monomers to be assembled in polymer chains of interest, natural and unnatural, Pistoia alliance, or custom libraries of interest. This is the best in class capability to make it easy as possible to diagram and create quickly these novel polymers where you can, select them and see what changes you have. And you can simply point and click, and Charles is able to build this out in this polymer sequence with these linkers quickly. So you'll notice briefly I'm hovering over things, or you saw some hovering over things. You'll get to see the sequence showing up and actual structure showing up. But also the chemical modifications that are added. So oftentimes you don't have a specialty like chemistry at the end, and the linkers need to be able to have it bound to the analyte and the payload. Okay, oops. So the value for Charles is really about being able to correctly represent the linker in a way that's consistent across the teams, from conjugation chemists, and across his organization, right? Because if he's working with many teams, as we've talked about, they may need to see it in their own, sequence or amino acid way, whereas he wants to see the chemical representation. And this supports this and provides the capability to draw and manage and standardize the complexity of these monomers required to represent these modalities. Okay, so finally, the team will get together to do the final assembly within Signals. They can also draw the antibody, or in this case, a segment of the antibody that's of interest. As I mentioned, the linker chemistries, as well as the linker to the payload. So to get an accurate representation of the final ADC that will go into testing. The team will then assemble the three main components, ADC, antibody, linker, payload, and this capability really brings it all together to keep all the components and the lineage and the data results connected is a very powerful and unique capability within Revvity to do the final assembly without losing any context. And I'll just mention quickly here, the key benefit for them is the full traceability, being able to, have all these individual components, all the individual results associated with them, and connect that to the final assembly that spans from small molecule to antibody to ADC, full ADC. And again, that's a unique capability that we support. Okay. So this brings us back to the beginning where I showed you the end dashboards. Here, not only do we have the antibody data, we have the small molecule linker, and all the sets are connected related to the ADC candidate. We have various dashboards of interest, a search where we can, select various aspects of interest and deep dive into this data. So once the search is performed, we can observe a dashboard that looks like the one I showed you in the beginning where all the data is connected. So the green is, well, once we get there, hold on. You can do it, video. Oh, maybe not. How interesting. Oh, there it goes. Okay, interesting. Pause for a second. So the green will be the final antibody-drug conjugate information or test results. The blue is the linker results for all the things like antibody load efficiency. The tan are the results related to the antibody itself. And the pink is related to the small molecule payload, which we'll show you in just a second. And, think for a moment that, what we're doing. This is being pulled, all those results are being pulled together, really disparate constituents and all part of a final single construct that are being assembled into a single dashboard that will, again, as I open it every time, will be refreshed with the correct data. So we can hover over this, pin a reference sequence, and see how these CDRs change between to get a snapshot view. Comparing payload versus ADC, looking at correlation analysis between the original payload test results is very nice. But I think the end goal really is to get to this final dashboard, which we'll show in just a moment. I previously showed you this highly multivariate space where you really want to see a tool that has the multi-parameter optimization. And this is considered a good result within the green, as I mentioned, for each of these axes. And it's intended to make it easy for a scientist to select ADCs and see how these ADCs are performing across, all of these different assays, various areas and parameters. And, in this case, we can quickly and clearly see that ADC 4162 is the winner. And that's, where we would want to move that forward, taking this very complicated multi-parameter optimization space and providing a visual reference space that makes it easy for data-driven decisions faster. So let's take a quick breather. Whew. Using signals, this ADC process can be driven rapidly and thoroughly, as I showed, end-to-end. And it's all happening within the Signals Research Suite, SaaS-based market-leading capabilities. So we continue to grow and improve and to add what Signals already offers today to make it as easy as possible for customers who are working in the antibody space to benefit from them. So this will end our in-presentation or demonstration part, and I will post the last poll question, and we'll give the next 30 to 45 seconds to participate from you all.
Last poll question is, what percentage of your experiments are outsourced? And the bars are moving. And the bars are still moving.
Lots of outsourcing going on.
All right, I believe we have stabilized, and the final answer is in. For percentages at the low end, 0 to 20%, that is 43% of the total from this group. In the 21 to 50% of the experiments outsourced, that is the answer selected by 36% of this group. 21% selected 51 to 80%.
Wow. Very nice. Okay.
Nobody is above 80% of this group.
That makes sense. Yeah. So depending on the audience, obviously that will strongly influence the outsourcing percentages, but it's important to note, again, we've taken you from like various different softwares that you might be using, various different internal groups that you might be using. And now let's add on another wrinkle where you have to outsource. So we're really thinking about it from end to end, trying to provide, a seamless experience for people. And obviously there are also, what did I want to say? Economic influences to that, but trying to make that process as seamless as possible so that your data is secured and it's generally a seamless experience between, sharing data, usually primary data sets, raw data sets between, a CRO or an academic collaborator, et cetera. So appreciate the participation there. All right, so I will move into the Q&A.